And the next vitamin in this series is folate, or it is also called as a folic acid. Okay, so this is folate refers to the class of compounds having chemical structure and nutritional activity very similar to folic acid or teroyl L-glutamic acid. So folic acid or folacin, they like, see, foliage or Latin word. There is a Latin word called folium. Folium means leaf. Okay. So from that this name has come. So it is abundantly found in green leaves. Okay, green leaves or vegetable or green leafy vegetable, and that is why the name comes like folium means leaf, and folic acid or folacin or folic acid, which is abundantly found in green leafy vegetable. Okay, so it is important for like one carbon metabolism. It is required for synthesis of amino acids such as purines and pyrimidine. Okay, so again, this is not just required for one specific function, but it is also required for synthesis of amino acids, purine, pyrimidine, thymine. Okay, pyrimidine and thymine. So what is its chemistry says? So folic acid consists of three components, like pteridine ring, aminobenzoic acid, that is PABA. This is this PABA, and then you have a glutamic acid. So it consists of three component, pteridine ring, that is this. Okay, and then you have aminobenzoic, para aminobenzoic acid, that is also called as a PABA. Okay, para aminobenzoic acid, PABA, and glutamic acid, like the folic acid mostly has one glutamic acid. Okay, only one glutamic acid residue, and is known as a teroyl glutamic acid, PGA. Okay, so the active form of folic acid is basically tetrahydrofolate. It is called as a tetrahydrofolate or FH4. Okay, tetrahydrofolate. It is synthesized from folic acid. Okay, this tetrahydrofolate is synthesized from the folic acid by the enzyme dihydrofolate reductase. Okay, dihydrofolate reductase. So you need this enzyme mm, for the synthesis of tetrahydrofolate, which is most active form of folic acid. The reducing equivalents provided by 2 mole of this NADPH. The hydrogen atoms are like, yeah. So basically, this is your folic. It is also called as this steroyl L-glutamic acid, and is coupled through this amino group to the carboxyl group of para aminobenzoic acid, which is in turn linked to a 2 amino 4 hydroxytannine. Okay, so this is major naturally occurring form of folate, and it is present in plant, animal, microbial sources also. They are present. Okay, so tetrahydrofolate, as I said, like THF or so relative to the nutritional needs of human, folate is frequently among the most limiting of the vitamin, because this is because of many reasons. Number one is poor diet selection, especially with respect to food rich in folate. For example, fruits, especially citrus fruit, green leafy vegetable, or because they are very strict vegetarian, they are not eating organ meats. Losses of folate, this is a one thing that I want to insist here. In India, the type of processing techniques that are being used, are very harsh, and we treat our vegetable at a high temperature, like nothing goes like no preparation goes without any high tadka, okay, high temperature heating of oil. And then even if you are cooking spinach or methi leaves or any leaves, we cook them so much. So these losses of folate occurs during this food processing, and home preparations are so harsh. We oxidize, and we also cause the leaching or sometimes oxidization and leaching of both, okay. Sometimes because of certain metabolic reaction, the incomplete bioavailability of folate also takes place. Many breakfast cereals like infant formulas, nutritional supplements, meal replacement, and also vitamin pill, they are fortified with folate. And Indian government or many countries, this folate or folic acid has been recommended to fortify along with staples such as whole wheat flour is consumed in India as in the form of chapati. So if one can fortify folate or folic acid, it will be a very good vehicle to supplement folate, because folic acid folate deficiency is one of the most common deficiencies seen, okay. So what is the stability and modes of degradation? Once we understand these, then only we will able to understand how much quantity one should fortify. So folic acid exhibits excellent retention during the processing and storage of fortified foods, and it is because of the water activity and premixes. Extensive losses occurs during processing, home preparation. Large losses naturally occur during the preparation and processing only. So this table indicates like how cooking can affect on folate content at a different form. Like for example, if you have broccoli, and 100 gram of fresh weight of the broccoli showed 169 milligram of folate, whereas the same broccoli's folate content was decreased to 65 when it was cooked, and the cooked water was analyzed, so the entirely it got leached into the cooked water. So cooked water was 116 milligram, okay. If you take the example of cabbage, almost 50% of the folate was lost in the cooking water. If you are using cooking water, then no problem, but otherwise there is a lot of 50% loss. Cauliflower, same thing, like it is almost everything like 56 to 47. Or from spinach, 143 milligram of folate was leached, 92 milligram, out of which 92 milligram was leached into the cooked water. So what is its degradation mechanism? The mechanism of folate degradation depends on basically form of vitamin, the chemical environment, whether in the surrounding the food that is being cooked, it has some other complex nutrient like presence of protein and acids and everything. So folate degradation generally involves changes at carbon 9 and N10 bond, the pteridine ring system, or sometimes both. So folic acid or THF folate and H2 folate can undergo the carbon 9 and N10 cleavage, and resulting into inactivation of the presence of either oxidants or inactivation in the presence of oxidants or reductants, okay. And that result into yielding of nutritionally inactive products such as para-amino-benzoyl glutamate, and a pterin ring was observed to be separated. So the most naturally occurring form of folate in many food is 5-methyl tetra folate form, okay, tetrahydrofolate form, that is THF or FH4. So most commonly the oxidative degradation of this form occurs by conversion to at least two products. So we have already studied any oxidation can result into formation of smaller degradation product, and then they further gets oxidized or degraded. So the first as 5-methyl 5,6 dihydrofolate, which retains vitamin activity, and then it can be readily reduced back to the 5-methyl tetrahydrofolate form by weak reductants such as thiols or ascorbate. So therefore, when food is rich in this vitamin, generally ascorbic acid or vitamin C can retain the degradation. So presence of other nutrients can prevent the degradation. So oxidative degradation of 10 formyl H4 folate can occur either by oxidation of the pteridine moiety to yield the 10 formyl folic acid. See, these steps of oxidations are like multi steps, and I have not included that because it's like a complex one, but understand that there is an oxidation of this, and there are a lot of factors that influence on the oxidative pathway in foods, and there are multiple like intrinsic factor, extrinsic factor, pH, complexity of the food that you are cooking. So factors that are affecting on folate stability, so to compare the relative stability of folates in the buffered solution as a function of pH, or as a function of oxygen concentration, or as a function of temperature. So it is a very complex, okay, stability of folates in food is very complex, and it is very less understood. So folic acid is generally most stable form that is considered, and therefore this folic acid is resistant to oxidation. Although reduced stability occur in the acidic media, like too much of acidic environment, H4 folate is the least stable form of the vitamin, or maximum stability of H4 folate is observed between the alkaline pH or pH beyond or between 8 and 12 and 1 and 2. So in extreme acidic condition it is stable, in highly alkaline condition it is stable, but if you see the pH below that range, it is not stable. So minimum stability is between pH 4 and 6. So it is highly pH dependent. Even in the favorable pH zone, it has been found in some cases of food. It found extremely unstable because it is not only one system like oxygen concentration, what type of form and what type of product it is, or food system it is there, whether there are other compounds that are present. So H4 folate having a substituent at N5 position exhibit much greater stability than doses unsubstituted with H4 folate. Okay, so this is because of the N5 methyl group is due, and at least it will take a part in the steric hindrance in restricting this access of oxygen or the attack of other oxidants onto this pteridine ring. Okay, so the stabilizing effect was observed in some of the products with respect to presence of this N5 substituent. Again, under low oxygen conditions or concentration, 5-methyl H4 folate and exhibit similar stability during thermal. So vacuum cooking or vacuum frying or vacuum processing maybe. So this is your tetrahydrofolate, and that gets reduced to N10 nitranium ion. Further Skips base are formed, and further it is forming 6-formyl tetrahydroprotein, and thus there is a degradation of your H4 folate. Okay, so these are all reactions. Oxidative degradation of this 10-formyl H4 folate takes place in presence of any oxidants. So what is the bioavailability of this folate in the food? The absorption of folate takes place mainly in the jejunum, and it is requires hydrolysis of polyglutamyl chain by a specific peptidase enzyme called as steroyl polyglutamate hydrolase enzyme. So this is followed by then absorption via carrier mediated transport process. Okay, there are multiple factors that are responsible for incomplete bioavailability, and that includes food matrix. As I said, whether you consume a spinach juice or raw spinach or cooked spinach. Okay, so presumably through non-covalent binding of folates take place with the other compound like protein, nitrogen, possible degradation of labile tetrahydrofolate form in the acidic gastric environment. Just now we saw that how this tetrahydrofolate is like unstable in the acidic environment. Incomplete intestinal enzymatic conversion of these polyglutamyl folates to the absorbable monoglutamyl form. So if the enzyme itself is not functioning, or compound that inhibit the steroid polyglutamate hydrolase absorption, or enzyme gets inactivated maybe because of the drug or some other compounds or the malabsorption of this. So fruits, vegetable, meats contain active hydrolases. Like along with the vitamin, there are also enzymes that are capable of deconjugating these polyglutamyl folates. Such processing methods such as homogenization, freezing, thawing, and other procedures that disturbs the cells may release this enzyme and promotes the deconjugation process. So how to analyze these folate enzyme, folate vitamins? One of the most difficult vitamin to identify or to quantify. There are microbiological methods such as HPLC method. Then there are competitive binding radioiso method. They could be used to quantify. In addition to that, most importantly, extraction of folate in an appropriate quantity is very important from the food sample, because we know that the food is a complex system. Sometimes intracellular cells, this is embedded, so their extraction via disruption of the food matrix is very important. But while disrupting, one should make sure that there less oxygen or less sunlight exposure, or while extraction itself the degradation should not take place, like cellular structure by homogenization in a buffer solution, or typically at 100 degrees Celsius, heat releases the folate from folate binding protein, and then to inactivate enzymes capable of catalyzing interconversion of folates to deproteinated form. Okay, centrifugation to yield a clarified extract, treatment with terol glutamate hydrolase or conjugase enzyme if the assay responds only to monoglutamyl. So there are various ways, and extraction is very important, and while extracting and reacting with the other enzyme, here should be taken care. In addition to that, microbiological growth assays are also traditionally followed. However, they take longer time, but they give very accurate, and these are very cost effective methods. For example, there are certain microorganisms whose nutritional requirement is only folate. For example, lactobacillus casei or pediococcus cerevisiae or streptococcus faecium or pediococcus cerevisiae and streptococcus faecium. So all these microorganisms will grow only in presence of folate or folic acid. So appropriate control of pH in the growth medium or this casei is equivalent response to all forms of folate. Maybe qualitative or quantitative also, you will have to like prick, like for example, if I take a plate and I take a 10 milligram or 5 milligram of folate, and if I say 10 or 100 colonies with 10 milligram, then I will say from spinach, if I take the extract and then I inoculate, and if lactobacillus casei grown on the plate are 200, that means there is a 20 milligram of folate present in the spinach. Okay, so likewise you can correlate. Since foods typically contain several folates, verification of equivalent response in microbiological assays is very essential. There are competitive binding assays which involves competition between folate in the sample or standard or with radio labeled folate for the binding site of a folate binding protein, especially from milk, can also be identified. What is the function of folate? The functional coenzyme form of folate is called as a tetrahydrofolic acid, like THFA, and this tetrahydrofolic acid is required in various functions like formation of adenine, guanine for DNA, formation of thymine for DNA by adding methylene to uracil. A vitamin B12 coenzyme is needed to recycle folate coenzyme for DNA synthesis. So, folate and B12 deficiency can produce identical signs and symptoms. Also in conversion of glycine to serine amino acid, that is called as amino acid metabolism, this vitamin is needed, or synthesis of neurotransmitter, very very important function. Cancer drug methotrexae this metabolism, which slows the cell division but results in anemia and diarrhea. Okay, so again this thing uracil, uh, and then thymine is formed, and then THFA transfer this single carbon group, that is from methyl, formyl, and methylene. So 400 microgram per day is required, and for recommended during pregnancy or lactation. However, daily requirement of folic acid could be around 200 microgram. Okay, so upper intake limit for synthetic folic acid is 1000 microgram per day. So higher than this may ask a vitamin B12 deficiency. Food processing can destroy 50 to even 100% of folate because of the heat, oxidation, and high temperature. So, if you look at the dietary sources, folic acid is widely distributed in nature. The rich sources of your green leafy vegetables, then you have these whole grains, cereals, liver, kidney, yeast, eggs, or milk is rather a poor source of folic acid, not a very good source. So if you look at this graph, cereal are very good source. Like if you take the whole cereal, almost 3/4 cup of consumption of whole cereal result into more than 100 percent of the recommended daily allowances. Whereas wheat checks is again whole wheat product can give you a very good requirement. Then orange juice also meets more than 110% of the recommended daily allowance, because orange juice is also very good source of these folate vitamin. Lentil, if you consume more than 75%. Okay, however, deficiency of this folic acid is probably the most common vitamin deficiency. You will be surprised, in spite of having folate distributed in so many common food, still we find this folic acid deficiency observed primarily mostly in the pregnant woman in both developed country as well as developing country, including India. India is the worst situation. The pregnant woman, lactating women on other diseases, and alcoholics are also susceptible to folate deficiency. Okay, the folic acid deficiency may be due to many reasons, like maybe it could be inadequate dietary intake or defective absorption itself of the folate or folic acid due to some wrong thing in the metabolism, or use of some of the anticonvulsant drug, like in depression or mental disorder. People take these anticonvulsant drugs like tone or dilatin or phenylalanine, and therefore there is an increased demand. Okay, so therefore in folic acid deficiency there is a decreased production of purine, and these purine and is observed, which impairs the we saw in one of the case, like in the DNA synthesis, this folate is required. So in case of deficiency, what happens, uh, there is a decreased production of purine, and therefore it impairs the DNA synthesis. And due to a block of DNA synthesis, due to there is a block in DNA synthesis, so there is no synthesis of the uh, DNA taking place. Okay, so this DNA synthesis, and due to block in DNA synthesis, the maturation of erythrocyte is slowed down, leading to formation of macrocystic RBCs or megaloblastic. This is also called as a megaloblastic. So RBC stays in immature form because there is no DNA synthesis in precursor cell is inhibited. Okay, so the rapidly dividing cells of bone marrow are also seriously affected. Okay, and that is why it is also called as a macrocystic anemia. Okay, the RBCs becomes abnormally large, or they are abnormally small. This is all associated with these megaloblastic changes in the bone marrow, and is a characteristic feature of this deficiency of folate. So, folic acid deficiency in pregnant women may cause like neural tube defect, it is called as a NTD. Okay, in the fetus. Okay, this is in the fetus. So therefore the pregnant women are given with the folic acid. Hence high doses of folic acid are always recommended for the pregnant women to prevent the birth defect related to neural tube defect in the fetus or infant. So folic acid is also associated with the metabolism of histidine. Okay, it is associated with the histidine metabolism. Histidine, or for me, this histidine is required in many other, metabolisms, and that has an impact in various other metabolic reaction. Yeah, so what happens, in there are steps in the folate deficiency. It is related to, megaloblastic anemia. So there are two types of anemia that are seen. One is iron deficiency anemia, and then another one is called as a megaloblastic. So this is the most common anemia that is observed uh, in India. Okay, so this is because of, you see, a decrease in blood folate concentration result into decrease in red cell folate and defective DNA synthesis, because DNA synthesis doesn't take place, a change in the structure of certain blood cells. So these are like normal blood cell in the bloodstream. The size, shape, color of the red blood cells show that they are normal. Okay, mature blood cells have like, see, but then you see here the megaloblastic blood cells seen here in the bone marrow are arrested at an immature stage of development. They still have their nuclei, see, in them, and are slightly larger. That is why it is called as a megaloblastic anemia. Okay. So in this, if you have a adequate amount of B12, cell divides normally. But what happens? Deficient people, the cells are unstable, and then they unstable to divide. Okay, so that is what is an increase in the size of circulating red blood cell and result into macrocytic anemia, or called as a megaloblastic anemia. And also, as I said, that it also impairs functioning of immune system. In developing embryos can cause neural tube defect in the fetus, and it's very very important for pre, it is also called as a prenatal vitamin. So this is a healthy spine. You see, this is the skin on the back, this is the spinal cord, and this is the spinal fluid, and this is the vertebra. But then this is because of the deficiency, the spine affected by the spina bifida. Okay, and then skin on the back, you see this neural tube defects that is connected to the spinal cord. So this is like a very very dangerous